this is Miss Ford again, and we're on round three with the light independent reaction of photosynthesis. So we did a part one, which was just an introduction. We did a part two, which was the light reaction, and now we're on the third video, which is the stage where glucose is finally made. All along, when we first started this, the reaction for photosynthesis included glucose. But so far, we haven't seen it yet. So, what we've done is looked at the stages that make the molecules that are going to be important for the light independent. And so, we are at the Calvin cycle. This took many years to kind of parse out just because of all of the reactions that take place, all of the enzymes, all of the molecules. Um, and it won a Nobel. Um, Sorry, it won recognition for Melvin Calvin to work throughout this. Now, there's three main stages, and this first one is pretty important. It's carbon fixation. It's going to be taking that carbon dioxide from the atmosphere and attaching it to a molecule that already exists within the chloroplast or the plant. And so we have carbon fixation, we have the reduction of our molecules, which are going to lead to our sugars, and then we have the rebuilding of the original molecule, which for a lot of students it's one of those things where they think, well, that seems wasteful. What's going on? Well, it's very interesting what why they have to do that. There's this molecule called ribulose bisphosphate. So that's RUBP, and it has 5 carbons. Now, I'm going to go ahead and start. Here we have 5 carbons. You'll see why I'm doing that in just a little bit. And we're going to start with 6 molecules. It's going to be an accounting. It's going to be nice to see why this occurs. Now, you might recall back in your chemistry days that reactions can be reversible. We oftentimes see A plus B yields C, but very often it can go the other way. C can break down into A and B. And what direction it goes is dependent upon a number of factors. The factor that we'd like to look at right now is the amount of reactants and the amount of products. If you have more of reactant A and more of reactant B, that's going to be those are going to be the molecules that are bumping into each other and colliding, and they're going to have the most likely chance of reacting. So the plant is going to make a lot of RUBP to have around to foster that direction. Let's get that carbon dioxide. Now, in a similar way, the plant would love to have lots of carbon dioxide. Now, that's going to become interesting question, because what if there isn't, or what if oxygen is piling up instead of carbon dioxide? But for now, let's just go with it. It'll be an interesting question later on. So the plant would love to have lots of CO2 and also lots of RUBP. And what that's going to do is drive that reaction forward. It's going to be a little more likely to happen. Now, it's still energetically unfavorable. Carbon dioxide just doesn't come out of the air and attach itself to things. So, what we need here is something that can attach 2 molecules together with a lower activation energy. Nice review point. Hopefully, you guys let your brain think for a second. Something that attaches 2 molecules together because they lower the activation energy and they increase the rate of reaction. Well, what we're thinking here would be an enzyme. And the enzyme is going to have an active site that's going to take carbon dioxide and then RUBP and then hold them in that active site and allow for that bonding to occur. So, what we end up having is a very brief, unstable intermediate that breaks down very quickly. Now, let's keep our numbers straight. We had 6 5-carbon molecules, that ends up being 30 carbons, and 6 carbon dioxides. Well, 30 and 6. What we have, very briefly, are 6 carbon molecules that break apart into 12 3 carbon. And if you're doing the math right, that should be 36. Yay. Um, this becomes important in a little bit because you want to keep track of where things are going. Now, Yay, we've grabbed carbon dioxide out of the air. Well, how do we change it to sugar? Well, we need energy, we need electrons, and we need enzymes to manipulate the molecules around. We're not going to worry about the enzymes right now, but they're there. They're converting things, but they need energy and they need electrons. So where are we getting that? Well, we're getting that from our light dependent reaction. So ATP, and then here we have our NADPH, our little truck that's carrying our electrons. He's going to swing by, drop off the electrons, yay, and we're back to NAD plus, which goes back to our light dependent reaction, just rotating back and forth that way. So the NADPH is going to drop off the electrons and hydrogens, or one hydrogen, and it's going to be used in converting those molecules. Now, you do not have to worry about the names of the molecules except for the end product. That end product is going to be one called glyceraldehyde 3 phosphate. Glyceraldehydes are a very simple sugar. Uh, Now, how did we get that? Well, we added electrons. Well, remember, that's a reduction. Leo says GER. So gain electron means reducing. So we've reduced it. Now, keep your numbers. We still have a 3-carbon molecule. It's been changed, but it's still 3 carbons. We have 12 3 carbons. We haven't lost anything yet. So we have a 3-carbon sugar, and 2 of them can then be pulled out of the reaction. 2G3Ps, glyceraldehyde 3 phosphates. Now, at this point, remember 2 3 carbons, that's a 6 carbon. This can now be converted to glucose. Glucose, a glucose and a fructose could be put together to make sucrose. We could put a whole bunch of glucose together to make starch. So the plant can do whatever it needs to at this point, make fructose for fruits, starches for the roots, whatever it needs. But the important thing is we've pulled it out of that equation. Now, at this point, a lot of students can look at this and go, well, now this seems wasteful because we're left with 10 G3P. What does the plant do but rebuild it back to RUBP? The remaining ATP from the light dependent reaction is being put back into this. Well, the reason we want that, remember, is the concentration. The more RUBP molecules you have, the better it's going to be to allow that reaction to go forward. So you've got a stockpile of these molecules, and when you use them, you got to rebuild them. So that's the goal of the RUBP. Now, what should you really be taking away from this? Well, certainly the energy requirements. Why do we use the enzyme? Why do we want lots of carbon dioxide? We want to take away the relationship between that and the light reaction. We want to take away getting our electrons and reducing it, and we want to certainly take away the purpose is pulling out the G3P to make sugars. You do not need to worry about all of these intermediates. That is not the goal. We don't need to memorize them. We do need to understand why and what the plant is doing with them. So hopefully that gets us thinking about some nice chemistry there.